Hi there, and welcome to a video outlining chromosomes and chromatids. Now, chromosomes are what we typically think of in our body cell and kind of in day-to-day operations, and they're what's called homologous. So we have one chromosome from the mother and one from the father. Now remember, in humans there are 23 of these pairs. I'm just drawing one pair that's here. These are called homologous. And they're homologous because they contain the same types of information. They're not exactly identical to each other, but the information they contain, let's say the information that's on there determines whether you have a widow's peak, attached earlobes, and there's actually a trait if your little pinky bends in. So maybe it's each of them have traits for that. Now, they may be a little bit different. Maybe one of them codes for widow's peak, one doesn't. Maybe one codes for your pinky bending in and the other one doesn't. Maybe both code for free or attached earlobes, but the general types of traits they code for are homologous. They are the same types of traits, but they may be different versions of them. So during mitosis, we need to replicate those, because the idea with cell division is that we're going to make copies so that we can then divide it again between the two donor cells. You're trying to make new cells. So in this case then, I'm going to try to keep my colors consistent here. Your first homologue would now become a pair of what we call sisters or sister chromatids. And then the other one would be the same. So I'm keeping the color kind of the same here. So same thing on both of those. Same thing. So they've made a copy of themselves. So let's zoom in a little on one of these sets of sisters. What's a little bit different, immediately you should see they're actually kind of connected here at the center, where when I drew them, and often they're shown this way in karyotypes and homologous pairs, because they're actually cut and pasted to kind of match up to each other. However, they actually are physically connected when they become the sister chromatids. And they're connected by a band around the region called the centromere. And I'm kind of exaggerating the size of the band here. And on the centromere, there's specific attachment points called kinetochores. And those kinetochores are extremely important in mitosis, because the idea with cell division is you're going to line up all of these copy pairs, these sisters, and then you're going to pull them to opposite sides of the cell. So when you pinch off or put your cleavage furrow down and make your two new cells, when you separate them, each side is going to get one of these. But they have to be kind of held together to be organized, and then they need to have that attachment point. This is where the spindle fiber is attached during mitosis. A couple other things to note here is when we look at this depiction with this kind of condensed X-like form, they are organized into pairs in general in the cell because we have those 23 pairs. But during mitosis, they actually do condense into a shorter piece, so it just gets smaller, if you will, and condenses, so that genetic information is in a more compact unit, so that way you can line them up across the middle of the cell and pull them apart during mitosis. And that's the basic structure, again, of a chromosome. So I'm referring to one of these here as just a chromosome. And then now when it's duplicated itself, I'm referring to those as sister chromatids. Yes, it does still contain the chromosome that's there, but now that we have a duplicated form, and again, this is seen during mitosis and meiosis as well, where you get this duplicated pair, we give it a different name because it's got that duplicated piece that's there. And so the idea is that we double the information prior to mitosis, so that when mitosis occurs and you do cytokinesis and split the cell apart, each cell, new daughter cell, ends up with a copy of each of those. I thank you very much, and please contact me with any questions.